If a trait is dominant, does that mean it is stronger, more common, or somehow better? No. And that single mistake causes a surprising number of genetics questions to go wrong. Dominant and recessive describe what you see in one particular gene combination. They are not rankings.
By the end of this, you will be able to read a Punnett square, explain Mendel's two main laws, and spot when the simple model does not apply. Start with the smallest useful vocabulary. A gene is a section of DNA that contains information used to make a functional product, often a protein. An allele is a version of a gene. For a simple classroom example, imagine a pea flower gene with one allele associated with purple flowers and another associated with white flowers. In diploid organisms, including pea plants and humans, you usually carry 2 copies of each gene, one inherited from each parent.
Your genotype is the allele combination you carry. Your phenotype is the observable result, such as purple flowers or white flowers. Genotype is the genetic recipe. Phenotype is what that recipe produces in a particular environment.
Now use the common symbols P and P. Capital and lowercase letters are a convention. The capital letter does not make an allele dominant. In this example, PP and PP both produce purple flowers, while PP produces white flowers. The purple allele is called dominant because its phenotype appears in the heterozygote, PP. The white allele is recessive because the white phenotype appears only when both alleles are the recessive version, PP. A genotype with 2 matching alleles, such as PP or PP, is homozygous. A genotype with 2 different alleles, such as PP, is heterozygous. Here is the key sentence. Dominant does not mean more powerful. It only means visible in the heterozygote for that trait. A dominant allele can be rare in a population. A recessive allele can be common. Neither label tells you whether a trait is helpful, harmful, normal, unusual, or more evolved. Gregor Mendel discovered the pattern by breeding pea plants in the 1850s and 1860s. He chose traits with clear alternative forms and began with true breeding plants, which repeatedly produced offspring with the same trait. For flower color, think of one true breeding purple parent as PP and one true breeding white parent as PP. Every offspring in that first generation receives a P from one parent and a P from the other. So every first-generation plant is PP and shows the dominant purple phenotype. Does that mean the white allele disappeared? No, it is still there in every PP plant. It is simply not visible in the phenotype. When Mendel crossed 2 of those PP plants, the hidden white phenotype returned in some offspring. Each PP parent can make 2 kinds of gametes, P gametes and P gametes. A gamete is a sex cell, such as a pollen cell or egg cell, that carries one allele from each gene pair. This separation of allele pairs into different gametes is Mendel's law of segregation. It happens because homologous chromosomes separate during meiosis, the cell division that makes gametes.
Each gamete gets 1 allele. Fertilization brings 2 alleles together again.
A Punnett square is a probability tool that tracks those possible gamete combinations. Put one parent's possible gametes across the top, P and P. Put the other parent's possible gametes down the side, P and P. Fill the boxes. You get PP, 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 and PP. That gives an expected genotype ratio of 1 PP, 2 PP, and 1 PP. Because PP and PP are both purple in this complete dominance example, the expected phenotype ratio is 3 purple to 1 white.
Expected is the important word. A Punnett square does not promise that every family of 4 offspring will contain exactly 3 purple and 1 white. It gives probabilities for each independent fertilization event. With many offspring, the observed numbers tend to move closer to the expected ratio. Think of each box as a fresh draw, not a prediction of birth order.
Here is a worked example you might see on a test. A purple-flowered plant has an unknown genotype. It could be PP or PP. You cross it with a white-flowered plant, which must be PP in this simple model. If every offspring is purple, the unknown parent was likely PP. If the offspring appear in an approximately 1 purple to 1 white ratio over many offspring, the unknown parent was PP. This kind of cross is called a test cross. It uses a recessive phenotype to reveal whether a dominant phenotype parent carries a hidden recessive allele. Mendel also proposed the law of independent assortment. In its simple form, it says that alleles of different genes can sort independently into gametes. For example, if one gene has A or A and another has B or B, an AABB individual can make AB, 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 and AB gametes. But do not use independent assortment blindly. Genes close together on the same chromosome are linked and are more likely to be inherited together. Independent assortment works best for genes on different chromosomes, or genes far apart enough on the same chromosome that crossing over separates them often.
Mendel's model is powerful because it is simple. Good biology also tells you where that simplicity ends.
Complete dominance is only one way alleles can interact. In incomplete dominance, the heterozygote has an intermediate phenotype. In a classic flower example, red and white alleles can produce pink heterozygous flowers. In codominance, both alleles are visibly expressed. The AB blood type is a well-known example. The A and B alleles are both expressed. That also reminds you that some genes have more than 2 alleles in the population, even though one person still carries only 2 copies. Many human traits are not Mendelian at all. Height, skin pigmentation, and many disease risks involve many genes plus environmental influences. These are polygenic traits, traits shaped by contributions from multiple genes. A simple Punnett square can model a single gene question beautifully. It cannot explain every complicated human characteristic. Now return to the word recessive. In some genetic conditions, a person with one typical allele and one recessive disease-associated allele has enough functional gene product to avoid the condition. That person may be called a carrier in an autosomal recessive inheritance pattern. But carrier, dominant, and recessive are descriptions of a specific gene and phenotype pattern. They are not labels for a person's value or health in general. If 2 carriers of the same autosomal recessive condition have a child, a simple Punnett square gives an expected 1 in 4 chance of an affected child, a 1 in 2 chance of a carrier child, and a 1 in 4 chance of a child with neither variant. Each pregnancy is independent. A previous outcome does not change the probabilities for the next pregnancy.
Real medical genetics can involve new variants, incomplete penetrance, variable expression, chromosome conditions, and environmental effects. A classroom inheritance pattern is useful for learning. It is not a diagnosis or a substitute for genetic counseling.
So why can one allele be dominant while another is recessive? Often one working allele produces enough functional protein for the usual phenotype to appear. In that case, a second altered allele may be needed before the phenotype changes noticeably. But dominance is not always about a working allele covering up a broken one. Some dominant variants create a protein with a new effect, interfere with the other protein, or change the amount of protein produced. The molecular explanation depends on the gene. Never assume every recessive allele is simply weak. Here are the 5 fast checks for any Mendelian genetics question. First, write what the letters mean and identify which phenotype is dominant in the stated model. Second, translate each visible phenotype into every genotype it could represent. Third, list the gametes each parent can make. Remember, each gamete gets 1 allele. Fourth, build the Punnett square or calculate the probability directly. Fifth, state whether your answer is genotype probability or phenotype probability. They are not always the same. Try one last question. 2 heterozygous parents, AA and AR, have a child. What is the chance of the recessive phenotype? Their possible offspring genotypes are AA, 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 and AA. Only AA has the recessive phenotype, so the answer is 1 out of 4, or 25%, for each pregnancy in this model.
Do not say 1 out of 4 children will definitely have it. Say each child has a 1 in 4 chance. That wording is scientifically correct and shows you understand probability. So, does dominant mean stronger, common, or better? No, it means that in a heterozygote, that allele's phenotype is the one you observe. Mendel gave you the basic pattern. Alleles separate into gametes, reunite at fertilization, and sometimes produce predictable ratios. Once you know the pattern and its limits, genes are no longer a code to memorize. They are a problem you can solve.